This afternoon, we want to look at gastrulation. Gastrulation focuses on what we call the trilaminar embryonic disc. Now, by definition, gastrulation is a process of formation of the three-layered embryo. And that three-layered embryo is what we call the gastrula. That is why the process of forming it is called gastrulation. We can call it the trilaminar embryo or the gastrula. I hope you do remember that from the second week of development, around day 9 to day 14, 15 there, we have a structure that is bilaminar. And so we call it the bilaminar embryo, with the two layers being epiblast and hypoblast. When the third week of development begins, or around there, we start to form a three-layered embryo. So a three-layered embryo forms from the two-layered embryo. So the process of forming this three-layered embryo is what we're calling gastrulation. The names of those three layers of this particular embryo are Ectoderm, Mesoderm, and Endoderm. So from epiblast and hypoblast, we are going to have Ectoderm, Mesoderm, and Endoderm. The process of forming the three-layered embryo takes place during the third week of development. Why is the third week of development such an important time during development of the human being? The third week of development is important because it sets the stage for the period of organogenesis. From our previous discussion, we divided the pre-embryonic, we divided the intrauterine developmental periods into three: the pre-embryonic period, embryonic period, and the fetal period of development. And importantly, we said that the events that take place during the first 2 weeks are the ones that fall under the pre-embryonic period. We've talked about those ones: first week of development and second week of development. The third week of development, therefore, is the beginning of the embryonic period of development. And the embryonic period of development is the period of organogenesis. So the third week of development is the one that is setting the stage for the development of these body organs. Occasionally, we will then refer to the third week of development as the week of threes. Maybe sometimes I feel it's just a matter of pushing it so much academically, because in the second week of development, we highlighted so many things that were occurring in twos. So perhaps we're just in some academic pressure to also then refer to the third week of development as the week of threes. But having demystified that, why do we call it the third week of, why do we call it the week of threes? We call the third week of development the week of threes because it is during this period that we have the three layers forming. We have already given names to those three layers: Ectoderm, Mesoderm, and Endoderm. Important to note is that those three layers all develop from the epiblast, that none of them actually arise from the hypoblast. It is the epiblast layer that gives us the three germ layers. The other thing why we call the third week of development the week of threes is because during this period, there are some three important embryonic structures which are formed. And these structures are the primitive streak, the notochord and the neurotube. The notochord really and the primitive streak are the ones which are formed during the 3rd week completely. The neurotube begins to form in the 3rd week, although it's not really complete by the time the 3rd week is ending. It is complete by the 4th week. But at least we still push that at least there's evidence of formation of those 3 in the 3rd week of development. In this lecture, we will talk about the primitive streak. The notochord and the neurotube, we'll talk about them in the next lecture when we talk about neurulation. The 3rd reason why we call the 3rd week of development the week of 3s is because during the 3rd week of development, the mesoderm layer, we said that there are 3 layers, ectoderm, mesoderm, and endoderm. That mesoderm layer forms some 3 segmentations. Those segmentations are known as the paraxial mesoderm, intermediate mesoderm, and the lateral plate mesoderm. Again, strictly speaking, these 3 layers are not necessarily completely segmented by the 3rd week, but at least there's evidence of that segmentation that begin from the 3rd week. And that's I was telling you, perhaps they're just trying to academically push for the 3rd week to be called the week of 3s. But either way, you've understood what I've told you. So what do we need to learn in this lecture where we're going to talk about gastrulation? We are going to first talk about how this primitive streak is formed. We are also going to see the functions of the primitive streak. And we are going to see the fate of the primitive streak. After that, we are going to describe the process, the series of events that take place during the gastrulation process when the 3 layers are forming. And then, importantly, we are going to name the derivatives of each of the 3 germ layers. And perhaps that's where the rubber meets the road. We are going to state the tissues, the body tissues, which come from each of the 3 germ layers. So let's start with the first agenda to talk about the primitive streak. What is the primitive streak? The primitive streak is formed by proliferation of cells of the epiblast. They're not just any random cells of the epiblast, but the cells of the epiblast which are at the center, the cells which are in the midline of the epiblast. These are the cells which proliferate. So if you look at this image, that particular region of the epiblast, remember this is the epiblast, this is hypoblast. So the middle of the epiblast, the cells at the midline of the epiblast are the ones which proliferate. That is if you appreciate it in cross-section. But if you appreciate it from the top, we note that yes, the structure called primitive streak is midline, but we also note something, that structure called primitive streak does not extend the whole of the epiblast. As a matter of fact, it is only on one end of the epiblast. That end of the epiblast demarcates the caudal end of the bilaminar embryo. And this will still be the caudal end of the baby. So the primitive streak forms on the caudal end of the baby, not the cranial end of the baby. And so that tells you that by the time the primitive streak has appeared, we know that this is the caudal end of the baby, and we know that this is the cranial end of the baby. I want you to note 2 things. That the epiblast is the one that gives you the primitive streak. I also want you to know the second thing, that the primitive streak is midline on the caudal end. It does not extend the whole of the embryo. So what are the parts of the primitive streak? There is a depression on the top of the epiblast, demarcating where the primitive streak is. That depression is called the primitive groove. It's a linear depression along the length of the primitive streak. Towards the end of that depression, there's a small swelling. That small swelling is called the primitive node. At the center of that node, at the center of that swelling, there is a depression that goes deep in form of a canal. We call it the primitive pit. So these are the three parts of the primitive streak. We have the primitive groove, which is that linear depression. Then the primitive node will be the swelling around here. The primitive pit is a deeper canal at the end or at the center of the primitive node. So those are the parts of the primitive streak. The evidence of formation of the primitive streak perhaps begin at around the 14th and the 15th day of development. Remember, we are counting from the day of conception. So I'm giving you the embryonic dating, not the gestational dating, which is counted from the last normal menstrual period. The two have a 2-week difference. Let's talk about the functions of the primitive streak. The primitive streak provides structural support to the bilaminar embryo. And I am underlining bilaminar here because we still have the epiblast and the hypoblast at this time. So it is providing structural support to the bilaminar embryo. But the presence of the primitive streak, now there is less flexibility. There is more rigidity being offered to the bilaminar embryo. And that's important. Apart from structural support, the primitive streak establishes the embryonic axis. We know that now this is the center of the baby. By the presence of the primitive streak, we know that this is the midline of the baby. We also know that this is the caudal end of the baby, and this is the cranial end of the baby. We know that this is the right side of the baby, and this is the left side of the baby. The primitive streak establishes the axis. And because the primitive streaks are the center, we now have the right and the left side being somewhat mirror image of each other. That's what I'm calling bilateral symmetry. Last but not least, the primitive streak direct the process of gastrulation. And that's why we are talking about primitive streak, because it is the structure that directs the process of gastrulation. After the primitive streak has performed its duties, and we are going to see how it performs that duty. But after the primitive streak has performed its duty, what happens? You and I don't have a primitive streak. There's a reason why. Because the primitive streak actually disappears. It is not uncommon to see embryonic structures appearing, performing their function, then disappearing. Primitive streak is just one of them. So after it has performed its duties, the primitive streak disappears. Now I want you to then ask yourself, what would happen if the cells don't disappear? Remember, the cells of the primitive streak are somewhat totipotent or pluripotent. They have the capacity of becoming multiple things, if not everything in the baby. And so these cells have the capacity of becoming different tissue lines, they are supposed to disappear. If they don't disappear, then they may grow into a tumor. Such tumors, we mentioned them earlier, we called them teratomas. Now, the unique thing about these teratomas is that the teratomas which are due to persistence of the primitive streak will be teratomas which will be on the sacral and coccygeal region of the baby, because we agreed that the primitive streak is on the caudal end. So they'll be in the sacral and coccygeal regions of the baby. We call them sacrococcygeal teratomas. As I told you earlier, teratomas have multiple tissue lines. So if you biopsy such a tumor, you may find bone tissue within it. You may find adipose tissue, as we commonly do find. You may also find other soft tissues, even elements of the skin like hair are still present there. It just tells you the totipotency or the multipotency of such tissues, of such cells, the cells that give rise to this tumor. Right, that's the story of the primitive streak. Now let's talk about the process of gastrulation. We have already said that gastrulation is the formation of the trilaminar germ disc. And we've called the trilaminar germ disc the gastrula. We have also indicated that, okay, this one we've not yet indicated, but I've already told you that this process takes place during the 3rd week of development. Now, how does it take place? The process of gastrulation occurs by migration of the cells of the primitive streak. The cells of the primitive streak, the cells which are formed, are the ones which migrate. Look at this image here. The blue represents epiblast layer. The yellow represents hypoblast layer. The middle here represents the cells of the primitive streak. These cells of the primitive streak migrate, but the migration is not a random one. The cells of the primitive streak migrate downwards, which means from here this way, towards the hypoblast, and outwards to mean from the center towards the peripheral side. So they migrate downwards and outwards. I prefer talking about this migration process in 2 phases to make sense for us. So let's consider that the migration process takes place in 2 phases or in 2 waves. In the first wave of migration, this is what happens. Yes, the cells of the primitive streak will migrate down and outwards. But as they do that, and especially when they are migrating outwards, they are displacing the hypoblast layer. So they displace and replace the hypoblast layer. This first wave of migration displace and replace the hypoblast layer. So in this image, the yellow cells are the ones which are doing that. The green cells represent the hypoblast. We can see the hypoblast being displaced laterally. The cells which replace and displace the hypoblast layer will constitute the endoderm layer of the gastrula. So this is the first layer that we are mentioning. This is the first layer of the gastrula that we are talking about, the endoderm layer. later on, we know that the hypoblast was covering the yolk sac, just like the epiblast is somewhat enclosing partially the amniotic sac. So because hypoblast was covering the yolk sac, and we are saying that we are replacing the hypoblast layer with the endodermal cells, it means that later on, the endoderm layer is the one that will be covering the yolk sac. So the cavity down here is the yolk sac cavity, just like the cavity up here is the amniotic cavity. So that is the first wave of migration. The point to note is that the cells displace and replace hypoblast, they become endoderm. The second wave of migration, this is how the cells behave. The cells migrate down, yes, and outwards, yes. But as they do that, they sandwich themselves between the endoderm that we've already established or that is still being established and the epiblast cells which are remaining. Remember, the primitive streak cells are actually epiblast cells. So the cells of the primitive streak have migrated, but there are some epiblast cells which are still remaining. So this second wave of migration, the cells sandwich themselves in between the endoderm that we've already established and the cells of the epiblast which are still remaining, the ones which have not yet migrated. That layer that is established in the middle of those two, the one that is sandwiched there, constitute the mesoderm layer. So this mesoderm here is different from the mesoderm we talked about during the second week of development. The mesoderm formed in the third week of development is a mesoderm that is within the embryo, the mesoderm that will form part of formation, that will participate in the formation of body organs. To distinguish this mesoderm from the mesoderm we mentioned in the second week of development, we call this mesoderm the intraembryonic mesoderm, which means mesoderm inside the embryo. To distinguish it from the mesoderm we talked about last week when we were talking about the second week of development, that mesoderm was called the extraembryonic mesoderm. If you do remember, the volume of cells that was filling the space between the derivatives of the inner cell mass and the derivatives of the outer cell mass. That was extraembryonic mesoderm. That appears in the second week. But here, this one, this intraembryonic mesoderm, appears in the third week of development. Later on, this is the mesoderm that is going to differentiate into three segments that I told you earlier: the paraxial mesoderm, the intermediate mesoderm, and the lateral plate mesoderm. We are going to see that shortly. So now we have discussed two waves of migration. The first wave of migration has seen that we have formed the endoderm layer. The second wave of migration has seen that we formed the intraembryonic mesoderm. There are still some cells of the epiblast which were not necessarily part of the primitive streak, and so they have not migrated. Those cells of the epiblast that remain, the ones that don't migrate, are the ones that are going to constitute the ectoderm layer. Therefore, the structure we have at this point has three layers: ectoderm, mesoderm, and endoderm. That's what we call the gastrula, the trilaminar germ disc, or the trilaminar embryonic disc. Important to note is that all the cells of the three layers have all come from the epiblast layer. None has come from hypoblast. So this image summarizes for you the process of gastrulation. First, we have the formation of the primitive streak. It forms in the midline and the caudal end of the baby. The cells of the primitive streak then migrate in two waves. The first wave of migration, we replace the hypoblast layer. The cells that replace hypoblast layer constitute the endoderm layer. The second wave of migration, the cells sandwich themselves between the epiblast that is remaining and the endoderm already formed. That layer constitutes the mesoderm layer or the intraembryonic mesoderm. The cells of the epiblast that remain constitute the ectoderm. And so at this point, we have a three-layered embryo. We're having ectoderm, mesoderm, and endoderm. This is the gastrula. And so that's the process of gastrulation. As I told you, the mesoderm layer is going to undergo some changes. As we can see from this end, followed by that end, all the way to here, we see that the mesoderm is going to undergo some changes. I'll take you through that shortly. Also, I want you to note that the ectoderm is also undergoing some morphological change. The morphological change that is occurring on the ectoderm is called neurulation. That neurulation is a process of formation of the neural tube. We will discuss it in our next lecture. But also see that the endoderm is also going some morphological change, largely because of change of morphology of the embryo, what we call embryonic folding. If you look at this embryo here, it's flattened, but as we follow this A, B, C, D, the D1 is almost curved, and even what we see on the other one. The embryo undergoes folding. I want to talk about the derivatives of each of the three layers, ectoderm, mesoderm, and endoderm, so that we see what each of the tissues become. Let's start with the derivatives of the ectoderm layer. Generally, ectoderm is important for protection as well as for communication. So even as we go through, think about those systems in the body which are for protection and communication. But this is what happens. After the three layers have been formed, ectoderm, mesoderm, and endoderm, still within the third week of development, there's a structure called the notochord that forms. This structure called the notochord forms within the center of the mesoderm layer. And it forms from the region of the primitive pit, cranially. So from the region of the primitive pit, cranially, that's where the notochord forms. Remember, it's lying within the mesoderm layer. Even though it is lying within the mesoderm layer, its molecular influence is actually on the ectoderm that is overlying it. Remember, the notochord is still at the center. And so it will have influence, the notochord will have influence on the ectoderm, which is immediately on top of it. This is what the notochord does to the ectoderm. The notochord induces the overlying ectoderm to differentiate into what we call the neuroectoderm. There'll be some molecular reasons why that is called so. But from a morphological point of view, the region we are calling neuroectoderm becomes a bit thicker compared to the regions that are not the neuroectoderm. So this region near the notochord, the region of the ectoderm near the notochord, is going to thicken. And then after that thickening, that region is going to fold on itself in the process to form a tube, what we call the neural tube. In simple terms, that region is going to undergo neurulation. That means that we are basically forming the nervous system. I don't want to dwell much into the process of neurulation and the result of neurulation, but this is what I want to say, that because of the presence of the notochord, the ectoderm differentiates into two parts. The neuroectoderm will give rise to the nervous system, both the central nervous system as well as the peripheral nervous system. And next time we are going to see that it is not even limited to the nervous system only. There are some cells which are neuroectodermal in origin, but they become other things outside the central nervous system. But for the sake of this lecture, let's just keep it simple to this extent at this point, that the region of the ectoderm near the notochord will become the nervous system, both central as well as peripheral nervous system. That means that there are some regions of the ectoderm which are away from the notochord, and in this image we can consider that part or that part to be away from the ectoderm, sorry, away from the notochord, the peripheral parts of the ectoderm, that part and that part. Look at what happened to them as neurulation is taking place, this part and that part. And by this time, the neurotube has formed, so this part that remains, see the way it's thin as drawn there. That part of the ectoderm is known as the surface ectoderm. The surface ectoderm becomes the epidermis of the skin. So we've addressed the concept of protection, epidermis of the skin, and communication, the nervous system. Those are the derivatives of the ectoderm. I want us to now talk about the endoderm layer before we talk about mesoderm. But for me to elaborately convince you on the derivatives of the endoderm, you need to see how the embryo folds, both in the transverse axis as well as in the sagittal axis. I will start with embryonic folding in the sagittal axis because that is more convincing on what I want to tell you. I'm going to play for you a video that shows you what happens as the embryo folds. In particular, pay attention to the endoderm layer. So let me pause it and tell you something. So this is the connecting stalk. So that reddish layer there is the splanchnic layer of the extraembryonic mesoderm. And this one here is the somatic layer of extraembryonic mesoderm. So this is the chorionic cavity. This is most likely the cytotrophoblast. And that other one is the syncytiotrophoblast. So the one that is giving us villus projections, that is cytotrophoblast. Then this is syncytiotrophoblast. The inner layer is the somatic layer of extraembryonic mesoderm. That's the connecting stalk, the future umbilical cord. This cavity there is the amniotic cavity, and the one down here is the yolk sac cavity. We see the trilaminar embryo there with ectoderm, the blue, endoderm, the yellow, and that thin red one, the intraembryonic mesoderm. So this is the trilaminar embryo, although we've cut it in a sagittal section, but that's a trilaminar embryo with ectoderm, mesoderm, and endoderm. You may not believe, but this is the region that will form the brain eventually. and perhaps that region, the spinal cord. And you may not believe that this is your future heart. We see at this point the heart is cranial to the brain, but in this folding, we are going to see what happens so that the heart actually becomes into the thorax. This is the yolk sac, and this is the, okay, we've not mentioned this before. This is called the allantois. We'll talk about it when we discuss the fetal membranes. Take note that this is the yolk sac and this is the amniotic sac. And because this is the yolk sac, this is the endoderm layer. I want you to see what happens to the yolk sac and the endoderm as the embryo falls in this particular axis, which we are calling sagittal embryonic folding. So now the embryo is beginning to fold. Look at that. Look at what happened to the dorsal part of the yolk sac. It is slowly being incorporated. Look at what's happened to the heart. The heart is descending down to the thorax and the brain becomes cranial. Yes, there's a part of the yolk sac that is still outside, yet there's also another part of the yolk sac which is inside. The part of the yolk sac that become incorporated into the baby as the embryo falls will become the primitive alimentary canal. We call it the primordial gut. So this is the primordial gut, the part of the embryo that become incorporated, the part of the yolk sac that become incorporated into the embryo during embryonic folding. As we can see from this image, it's only the dorsal part of the yolk sac that is incorporated. The ventral part of the yolk sac remains outside. The connection between the two is called the vitelline duct. We will revisit this folding again when we talk about fetal membrane. So for now, the take-home message that I want you to get, at least at this point, is that at some point the embryo will fold. And when the embryo folds, the yolk sac become incorporated into the embryo. And that yolk sac represents the future alimentary canal, what we call the primordial gut. So these images show us that concept of the primordial gut being incorporated into the embryo as the embryo falls. So having said that, what will the endoderm become? So this is the endoderm that is lining the primordial gut, basically. It is the one inside the primordial gut. The endoderm layer is largely for nourishment. It gives rise to the epithelial lining and glands of the digestive and respiratory system. It gives rise to the epithelial lining and glands of digestive and respiratory systems. Those are the derivatives of the endoderm. Remember, it's inside, so it will give rise to the epithelial lining. But glands also come from the epithelium, so it's okay for them to give rise. Now, why respiratory system? Because respiratory system is basically a diverticulum from the digestive system. That is why air and food have a common channel at some point. The throat is a common channel for food and air. The lower respiratory tree actually is a diverticulum from the digestive system. That's why it's possible to choke. Food can go to the windpipe instead of going to the food pipe. All right, so that's endoderm layer. It forms the epithelial lining and glands of digestive and respiratory system. I want to now show you embryonic folding in the transverse axis so that you still appreciate the concept of endoderm being incorporated into the baby as the baby falls, but also to appreciate something else, how the mesoderm segments into 3 parts, and also how the neurotube forms. Perhaps I will play this video in I'll play it 2 times. The first time, let me focus it on showing you what happens to the what happens to the ectoderm and what happens to the yolk sac. Then the second time I'll play it, I'll show you what happens to the mesoderm. So I'm playing first round. We can see we have the ectoderm, mesoderm, endoderm. The green is an autocode. Now see how the embryo, first of all, we've not yet started folding, but see what's happening to the ectoderm. The ectoderm is folding on itself. That process of folding of the ectoderm is what we are calling neurulation process. The region near the notochord are the ones which are undergoing neurulation. So that part of the ectoderm that undergo neurulation will form the neurotube, the one I'm pointing at. But also see how the baby is now curving, and importantly, see how the yolk sac has become small, with the dorsal part of the yolk sac being incorporated into the baby and the ventral part of the yolk sac remaining outside. See how the shape of the baby has changed. In cross-section, the baby is now cylindrical from a flat structure. It explains to you why you and I are cylindrical beings as opposed to just being a flat structure like tapeworm. All right, I promised you I'll play it 2 times. Now let me play it the second time. This time round, I want to focus on the mesoderm. So as development continues, the mesoderm layer differentiates into 3. See how those 3 are becoming at this point. There's this part of the mesoderm very near the notochord. That's what you call the paraxial mesoderm. So that and that are paraxial mesoderm. Then there's this part of the mesoderm in the middle. We call them intermediate mesoderm. Then there's this part of the mesoderm that is peripheral. We call them the lateral plate mesoderm. The lateral plate mesoderm is later going to divide into 2, as we can see here already happening. There's the part of the mesoderm near the endoderm. That part of the mesoderm near the endoderm is called the splanchnic layer of the intraembryonic mesoderm. And then this other one is called the somatic layer of the intraembryonic mesoderm. The somatic layer of the intraembryonic mesoderm is also called the parietal layer of the intraembryonic mesoderm. And this splanchnic layer is also called the visceral layer. But simpler terms we give to them is this splanchnic layer of the lateral plate mesoderm is also called the splanchnic mesoderm. And then this somatic layer of the lateral plate mesoderm is also called the somatic mesoderm. So let me finish that video. See how the mesoderm is divided into 3 and see the 2 layers of the lateral plate mesoderm. The splanchnic layer of the lateral plate mesoderm, you can just call it splanchnic mesoderm, and the somatic layer of the lateral plate mesoderm, you can just call it the somatic mesoderm. Also see what's happening to the yolk sac now. The yolk sac becomes smaller. The embryo is folding to become cylindrical. Some part of the yolk sac become incorporated into the baby to become the primordial gut or the primitive alimentary canal. So this is the part of the yolk sac that is incorporated. So having talked about that, we can now talk about the derivatives of the mesoderm layer. These are still images showing us what we've already described. And so because I don't want to repeat much, I think I can just go direct to the derivatives of each part. But remember, this is the paraxial mesoderm, this is the intermediate mesoderm, and we have two layers of the lateral plate mesoderm, somatic mesoderm and splanchnic mesoderm. Let's start with the paraxial mesoderm. What are the derivatives of the paraxial mesoderm? The paraxial mesoderm undergoes segmentation. First of all, why are we calling it paraxial mesoderm? Because it's the mesoderm parallel to the body axis. It is the mesoderm near the notochord. So this paraxial mesoderm undergoes segmentation. It forms some things which look like cubes on either side of the notochord. Those cube-like things are called the somites. The somites form in a very sequential, progressive manner, so that there's a time that you have so number of somites, then after a few days you have so number of somites, until the maximum number of somites are reached. We can therefore tell the age of the baby based on the number of somites which are present, but that kind of embryology is beyond this scope. So the understanding is that the somites are forming on either side of the notochord in a sequential manner according to the number of days until the maximum is reached. So these are the somites, cube-like structures on either side. Each somite differentiates into two things initially. These are the two things. So this is the somite. The somite differentiates into two things. There's what we call the sclerotome. The sclerotome is a derivative of the somite. And this sclerotome usually will form around the notochord and the neurotube that has already formed now. That sclerotome gives rise to the axial skeleton. If you remember, we know the components of the axial skeleton, the vertebral column, the cranium, the ribs, and the like. So this is where the axial skeleton comes from, the sclerotome. The second component of the somite is called dermamyotome or dermomyotome. But this dermomyotome, which is this structure here, immediately divides into two. We have the dermatome and the myotome. The myotome is the one that gives rise to the skeletal musculature. So think about skeletal muscles wherever they are. They come from myotome. And then the other component, dermatome, is the one that becomes the dermis of the skin of the trunk. The dermis of the skin of the trunk, the skin of the trunk, and especially the skin on the back of the person. That dermatome gives rise to the dermis of the skin. Or let me put it this way, the dermis of the skin of the back come from dermatome. That should trigger some unanswered questions yet. The unanswered questions that should trigger should be, so where do the other types of dermis, where do the other dermis and the other parts of the body come from? You should be asking yourself that question even as you go along. But let's talk about the intermediate mesoderm. This is the intermediate mesoderm. The intermediate mesoderm gives rise to the organs of the urinary system as well as organs of the reproductive system. So think of kidneys and ureter. Think of uterus, think of testes, think of ovary, they come from the intermediate mesoderm. Then we have the somatic mesoderm. Somatic mesoderm is this mesoderm that is near the surface ectoderm. Somatic mesoderm gives rise to the appendicular skeleton. Remember, appendicular skeleton refers to the skeleton of the limbs. So bones like femur and tibia would most likely then arise from the somatic mesoderm. Apart from that, somatic mesoderm also gives rise to the dermis of the skin of the anterolateral body wall, as well as the dermis of the skin within the limbs. The dermis of the skin of the anterolateral body wall, as well as the dermis of the skin within the limbs. Lastly, the somatic mesoderm also gives rise to the outer layer of the body membranes, the outer layer of the mesothelia. Remember, we have three types of mesothelia. We have the pleura, we have the pericardium, and we have the peritoneum. The outer layer of those three are the ones we call the parietal layer. So the parietal layer of the pleura, parietal layer of the pericardium, and the parietal layer of the peritoneum. The outer layer of the mesothelia come from somatic layer of the lateral plate mesoderm. Lastly, we have the splanchnic mesoderm. The splanchnic mesoderm is intimate with the endoderm, which means it is intimate with the primordial gut, because this is the part that will become the primordial gut. Splanchnic mesoderm gives rise to the smooth musculature. Could be the smooth muscles within the digestive system, smooth muscles within the respiratory system. It also gives rise to the cardiac musculature. We know that that one is found within the heart wall. Splanchnic mesoderm also gives rise to the connective tissue of the respiratory and digestive system. Connective tissues of the respiratory and digestive system come from splanchnic mesoderm. Lastly, the splanchnic mesoderm gives you the visceral layer of the mesothelia. So it could be the visceral layer of the pericardium, visceral layer of the pleura, and visceral layer of the peritoneum. Those mesothelial layers come from splanchnic mesoderm. Great. So I've given you derivatives of each of the germ layers. You may not believe, but by this lecture, we've accounted for almost every organ in the body. Think of any organ, we've most likely stated where it comes from. All the organs in the body. So this is one of those lectures that you must again listen to again, read more and more, so that you understand. I can just put it randomly right now and ask you, so those cartilages which are on the wall of the trachea, where do they come from? And you should be able to answer that question. I can pop it to you. How about the liver? So where does it come from? In one way or another, I've answered that question. I've told you that the endoderm gives you epithelial lining and glands of the digestive system. If you consider the liver to be one of those, then you have your answer. I've told you that splanchnic mesoderm gives rise to the connective tissue of respiratory and digestive system. If you consider cartilage of the trachea to be connective tissue of respiratory system, then you do have your answer. So every organ in the body, every tissue in the body, we have accounted for, and none have we not accounted for. But you have to revisit this and listen to again and again, or perhaps just read more about it so that you understand the derivatives of the germ layers. But importantly, if you notice in that lecture, we've talked about 3 layers: ectoderm, mesoderm, and endoderm. And I've told you many things about them. But one layer strikes out. The mesoderm gives rise to many things in the body. And because mesoderm gives rise to many things in the body, sometimes you may have a deficiency of mesoderm. And especially the fact that we need mesoderm to even migrate to some regions where it is not found. If we have deficiency of mesoderm, as it can happen sometimes, it might be a trouble, and a big one for that matter. In most cases, the lower parts of the baby are the ones which will suffer more. And so what happens? If the lower parts of the baby are deficient of mesoderm, you're going to have malformations which look like the ones projected there. The lower parts of the baby will be underdeveloped. Maybe they only have 1 limb, lower limb, instead of 2. Maybe they don't even have an anal opening. Maybe the reproductive systems are not even there. You can't even tell whether this is male or female. Maybe even the sacrum is affected. We don't have an imaging of that, but we can make that assumption. So it's a constellation of malformation. It's a syndrome. We commonly call it caudal dysgenesis, or because it's syndromic, we call it caudal regression syndrome. Caudal regression syndrome. It's a spectrum of malformation. It could just be as mild as everything is normal except that the baby doesn't have a sacrum, to the extreme that there's no even 1 limb, or the 2 limbs are fused. Right, so I am now done with the lecture on gastrulation. Your next lecture will be on neurulation, not today, but another day. I will now invite you for questions if you do have.